recording, and we switch over to Marta Borowska, who is now at Stanford. And brief bio on Marta: she joined the University of Chicago as a PhD candidate to pair her interests in structural biology and immunology. And now she continues to use this foundation as a springboard for engineering designer approaches in immunotherapy at Stanford University. And we're looking forward to your talk on polyreactive antibodies.
Thank you, Victor. I hope everyone can see my slides and hear
Yes.
me well.
Yes.
Yes, I'm currently a postdoc in Chris Garcia lab. However, today I'll be presenting part of my graduate work I did at UChicago in Erin Adams' lab. But I'm currently also working on an interesting topic that involves antibodies and antigen interactions. And I'll be happy to come back if you'll have me next year once I'm ready with my current data. But for now, I will dive in a bit more molecularly in depth into a nice case of natural polyreactivity antibodies. And everyone here is, I'm sure, familiar with the structure of the following antibody, but my point here is that since I'm looking at the molecular details of this antigen-antibody interaction, I will be referring only to this monovalent antigen-binding fragment, or a Fab, for the rest of my talk. So I will discuss a case of polyreactive antibodies, which are antibodies binding to this broad repertoire of antigens. And the major difference between polyreactive and monoreactive antibodies is that a single polyreactive antibody is able to bind to multiple diverse ligands as opposed to one ligand. And this binding is characterized by a very weak affinity. So this high specificity of monoreactive antibodies and autoimmune receptors is the basis of every antigen-antibody interaction, but it comes at a cost, since every interaction in biology is related to these fundamentally important issues. And one of them is the uncertainty about the target, which relates to a high level of target possibilities in terms of their sequence and structure. Another reason is the finite capacity of the recognition, meaning we only have a limited number of receptors that are produced in our body that can recognize distinct epitopes. Additional issue is that the noise in the recognition process is pretty high, which means that even with like a perfect pair of receptor and epitope pairing, the recognition and activation might not still take place. So a possible solution to be able to accommodate all these different numbers of targets is by promoting a molecular overlap among receptors, which is often refers to promiscuity or polyreactivity in B-cell receptors or cross-reactivity in T-cell receptors. So promiscuity is inherent and very common property of recognition among many immune molecules, including TCRs, recognizing different arrays of peptide MHCs that leads to a range of different responses. So polyreactivity is a common feature of B cells, with polyreactive B cells comprising about 6% of the naive B cell repertoire and about 25% of the IgG memory B cell pool. So it's proposed to overcome this extreme antigenic variation that PCRs may have evolved to harbor polyreactivity to adapt to ever mutating and shielded by glycosylations, hard to reach epitopes. However, the precise role of this polyreactivity in B cells still remains unclear. So how do we define polyreactive antibodies? So in case of the high affinity monoreactive antibodies, the goal is to bind to a single target, in example, the viral receptors, like we learned in the previous talk, with high affinity and high specificity. Then there are these broadly neutralizing antibodies or cross-reactive antibodies that can recognize the same target epitope, but of multiple strains of the same virus. So these antibodies are the holy grail of the universal vaccine for viruses like flu, HIV, or coronavirus. But interestingly, broadly neutralizing antibodies against HIV and influenza, and I'm guessing soon the research will show also the case for coronavirus, these antibodies are very commonly polyreactive because nearly 60 to of broadly neutralizing antibodies, besides recognizing their high affinity viral target, they also recognize other diverse epitopes, but with lower affinity. So it is hypothesized that polyreactivity can augment the overall binding strength to these viral epitopes by utilizing the binding surroundings for increased avidity, or by accommodating the molecular crowding at the membrane, surface charges, glycosylations, and other things that receptors have to do in order to search for its epitope. So the additional studied case of polyreactive antibodies are also the intestinal or homeostatic IgAs. They coat a variety of commensal microbiota, and they've been found to be naturally polyreactive. But in contrast to these antiviral IgGs, which also derive from adaptive immune system, these homeostatic IgA antibodies, they do not have a known high affinity ligand. So this poses an interesting question about the origin of polyreactivity and whether it is acquired through affinity maturation, or there are polyreactive naive B cells that are just simply recruited into this antigen antibody responses. So a few questions come to my mind when studying polyreactivity. First one being, what is the binding mechanism utilized for different antigens? And some factors being proposed so far to be involved in polyreactivity include the length of the CDR3 loops, net hydrophobicity or charge. Other studies narrowed it down to an increased prevalence of certain residues like arginine or tyrosine in polyreactive antibodies in their CDRs. Another aspect interesting to study is what is the structural basis for binding of polyreactive antibodies. So in the past, these high affinity antigen-antibody interactions have been thought in terms of a lock and key hypothesis, where antigen and antigen binding pocket acts as this rigid, perfectly complementary pieces. But this theory has been challenged multiple times since. However, it's been proposed that polyreactive or germline-like antibodies are more flexible than the classic monoreactive or antigen experience antibodies. And this flexibility was proposed to allow them to accommodate these diverse ligands within a single binding pocket. And lastly, what are the affinities of these polyreactive antibodies toward different ligands? So I investigated a panel of polyreactive antibodies that were derived from single cell derived antibodies that are routinely screened through this antigen binding promiscuity assays. We obtained these antibodies from Jeff Bunker from Albert Bendelak lab at UChicago, who screened through 1,000 of mouse IgA antibodies that were derived from the gut using ELISA assay. This ELISA assay uses up to 7 varieties of structurally and biochemically distinct antigens that include DNA, insulin, LPS, flagellin, albumin, the standard set of diverse antigens. And they represent a diverse sampling of polar, amphipathic, large or small-sized molecules. And out of these thousands ELISAs, he identified 6 most polyreactive clones that we decided to focus on in terms of studying polyreactivity in more molecular details. So here I'm showing you examples of 3 of them showing pretty high reactivity toward all 3 ligands. So in order to study other molecules that would be better targets for crystallization studies, we expanded these panels toward other targets like lysozyme, ubiquitin, and some pieces of single-stranded DNA. And we've observed for all of these polyreactive antibodies that we are obtaining the similar level of binding for these new ligands as well as the previous panel. So the low affinity interactions are very difficult to detect by standard biochemistry techniques, especially when measuring direct monomeric binding events rather than the full-length antibody. Therefore, my attempts to form complexes between these ligands and polyreactive antibodies by size exclusion were unsuccessful, and my studies by biolayer interferometry were also quite challenging. So no binding was observed between the polyreactive Fabs and lysozyme and ubiquitin, or even a dimeric ubiquitin ligands using this method. However, I was able to observe binding to biotin immobilize short single-stranded DNA pieces with all these 6 different polyreactive mouse Fab clones. And the range of affinities was detected between 30 to even 100 micromolar, which, to put it in context, these KDs are similar to those measured for conventional alpha-beta TCRs to their cognate ligands that are presented by MHC. So next, we were interested in what residues in polyreactive antibodies are involved in binding to these diverse ligands. So we were able to mutate every position in each of the CDR loops of heavy and light chain for these polyreactive antibodies. And we were able to see a reduction, which is shown in red, orange, and yellow in binding for specific residues. And previously, the reports were suggesting specific inclusion of arginine or tyrosines, or only a heavy chain or long CDR3 loops in mediating the polyreactive interaction. However, as you can see in the 6 cases, I'm showing you 3 different polyreactive cases, we haven't seen any patterns. Like in case of this mouse antibody, heavy chain is heavily involved, but in case of this antibody, it's the light chain heavily involved, as well as no particular residues seem to be abrogating the binding. However, in all cases, a single mutation would disrupt binding to all of the ligands rather than spread across different ligands. So many conventional sequence-based comparisons between polyreactive and monoreactive antibodies were quite unsuccessful, although we've dabbled with it a little bit as well using machine learning. And we searched for clues in the structures of the polyreactive antibodies, which I mentioned before. Some of these are the polyreactive mouse IgAs, but we've also included a human anti-flu broadly neutralizing antibodies that we obtained from our collaborators, Patrick Wilson lab at UChicago. And we are showing here. So I solved a total of 5 high-resolution structures, 3 on the left of polyreactive Fabs and 2 of the monoreactive by X-ray crystallography. And interestingly, the Fab structures range from having, short CDRs forming a flat antigen-binding surface, like in case of the 33-8E6 or 3BO3, to longer CDRs extending the antigen-binding side, like 43G10 to G02 or the 4CO5. So interestingly, the CDR3 of the heavy chain on monoreactive 4CO5 is forming this ribbon, kind of a structured extension of the CDR3 loop. And to investigate previous suggestions that polyreactive Fabs have overwhelmingly positive antigen binding side due to this prevalence of lysine and arginines on the surface, I inspected the binding side of the crystal structures. I solved for any hydrophobic on the bottom or electrostatic on the top patches in the surface view. However, I've not observed any predominant patterns that was present in terms of the charge and hydrophobicity when compared between poly and monoreactive Fabs. So neither was any pattern observed in additional structures I've looked at that were taken from the structure database of the anti-flu antibody. So this is in agreement with our single alanine mutagenesis, the ELISA I was showing before, when there's no pattern between the clones. And in conclusion, polyreactivity cannot be simply determined by just looking at the sequence or a static structure. Therefore, I resorted to using a dynamic studies, using the solved structures. So as I mentioned in the introduction, like multiple groups reported that polyreactive antibodies are more flexible as opposed to the rigid nature of monoreactive antibodies. And these studies were limited to only studying the CDR3 loops of the heavy chain or a single source of polyreactive antibodies, or they would rely on in silico predicted models of the structure, which the computational methods are still, they need a little bit more work in terms of predicting structures of flexible loops. So I have therefore explored a more comprehensive molecular dynamic analysis of polyreactive and monoreactive Fabs using these crystal structures and analyzing all CDR loops. And together with my colleague from Adam's lab, Chris Bochter, we run all atom molecular dynamic simulations using different architectures for an extended 500 nanosecond time. And our analysis follows this workflow for each Fab where we use a time-independent component analysis, which is able to reduce the dimensionality of our simulations, and this way sort all different structural conformations of a simulated Fab into multiple clusters from here. We further pick random 5 clusters by dropping a pin in the center of it and visually sampling representative conformations from these clusters. And then additionally, we expect root mean square fluctuation, RMSF, of the backbone and sidechain movements of these individual CDRs to observe how long these different conformations persist. So in our dynamic studies, we observed that structures of polyreactive Fabs are overall quite rigid compared to monoreactive Fabs. So the top would represent a case of a polyreactive antibody. Let's focus on that first. So TICA analysis for the polyreactive Fabs show consistently less clusters of distinct conformations, suggesting overall more static behavior. Occasionally, there will be like a global shift present in these Fabs, but overall shape of the loops is conserved, which would suggest that maybe the original state that was captured in a crystal structure might be different from the relaxed version of the protein in solution. So on the contrary, the monoreactive Fab on the bottom here, the 3BO3, shows more distinct CDR conformations and higher RMSF in the heavy chain in this case. The CDR2 of the heavy chain of the 3BO3 adopts distinct conformation and many events of exchange between these conformations as opposed to the polyreactive 2GO2. So this is especially intriguing observation given that both Fabs evolved toward flu target and use CDRH3 to mediate this high affinity interaction. So these dynamic differences, although relating to selected CDR loops, only reveal overall a more rigid nature of antigen binding surface in polyreactive antibodies. Although currently this data does not directly explain how polyreactive ligands would bind to the surface, because we don't know where polyreactive antigens are binding. However, I hypothesize that this rigidity can play a supporting role in polyreactivity. In example, you could imagine a firm binding surface could serve as like a uniform platform that is broadly permissive for different types of polyreactive ligands. And lastly, I further investigated the molecular basis of this rigidity for specific CDRs in polyreactive antibodies and compared them with increased flexibility in monoreactive Fabs. So I was specifically interested in like how this extended CDR, one of the light chain in polyreactive antibody on the left maintains its rigidity as shown by RMSF despite no stabilizing secondary structures, and how this CDR3 of the heavy chain in monoreactive antibody shows flexibility despite forming this structured beta ribbon formation. So upon closer inspection of these intra-chain interactions of these two CDRs, both showed a striking hydrogen bond network mediated by these CDR residues, but in two different ways. So in case of these long unstructured CDR1 in polyreactive antibody, it's extensively supported by multiple hydrogen bonds that are mediated by side chains, which together form this three-dimensional stable network. But in contrast, the structured CDR3 of monoreactive antibody forms also vast hydrogen bonds. However, it's mostly mediated by backbones of the ribbon and only allows for the two-dimensional stability. So these are just the two cases, but this raises an interesting hypothesis whether, in case of the polyreactive antibodies, this loop, the CDR1 loop, could possibly shield any polar or hydrophobic residues from intercepting the binding by engaging its side chains for stability rather than extending outside. So in this case, side chains, in the case of the monoreactive antibodies, the side chains could be exposed and sort of like clash with all the promiscuous ligands that will be able to approach. And I further look for other examples of rigidity in the CDRs in polyreactive antibodies and notice that mostly the side chains also mediated the network of that stabilize the CDR3 loop of the heavy chain of the polyreactive 2GO2 antibody. So here I'm showing 7 hydrogen bonds that stabilize this loop with positively charged arginine 100 that is facing inward of the loop. And interestingly, the same arginine 100 when they are both, and together with the arginine 101 position, when they are both mutated to lysine, so a very subtle mutation, they do show reduced polyreactivity, which consistently with the hypothesis in our dynamic studies, this double mutation could come, introducing flexibility by introducing these lysine mutations could come from losing this guanidinium group on arginine that could stabilize this three-dimensional network in exchange for more two-dimensional ammonium group from lysines. So this subtle little mutation could add up in overall maintaining of the rigidity in polyreactive antibodies. So to sum it up, I was able to determine the affinity of multiple polyreactive Fabs to one, unfortunately just one ligand, which is on the low end of protein-protein interactions. But considering that antibodies have increased valencies, they can form higher order complexes and add to the overall final avidity of polyreactive interaction. And I was able to show through my dynamics work on polyreactive Fabs that we noticed that there's a tendency toward rigidity for polyreactive antibodies and less conformations adopted by the CDRs of polyreactive antibodies. And yeah, there's many interesting follow-up questions that would require getting a structure of a polyreactive antibody with polyreactive ligands to be able to determine what is the actual binding sites of these ligands. And yeah, with that, I would like to thank my graduate lab, Erin Adams' lab. Everyone's been absolutely wonderful. And of course, Chris Bochter for his pretty much side-by-side work on this project. And also our collaborators from Patrick Wilson and Albert Bendelak in providing their expertise in polyreactive antibodies and broadly neutralizing antibodies. Thank you.
Thank you, Marta. That was really beautiful talk. I'm sure that there are questions. I'm going to start with a general question again. Can you hypothesize on how many polyreactive antibodies we have at any one time and as part of the B-cell receptor repertoire that would fit for your definitions? Kind of what's the percentage you think?
Yeah, so there is some data that shows that from the naive repertoire of B cells, it's about 6% of them. And on the memory pool, it's about 25. So it definitely increases over time, probably as we experience. I don't exactly know how it was quantified. It does seem a little bit high. But on the other side, like just knowing how promiscuous the immune receptors are and how important this feature is, I think that that's not really that surprising.
Mm. Mm. And then when you say with polyreactivity, rigidity increases or vice versa, that's also people say when from going from IgM to IgG and with affinity maturation, the antibody becomes more rigid. So that, and with the numbers that you said previously, that the memory compartment has more polyreactive than the naive. So that would kind of maybe, yeah, which is surprising because people always say as you affinity mature, you become more specific, whatever that means, but that, yeah, it's almost the other way around, or sometimes the other way around. Yeah. So it's a more complex picture. Yeah.
For sure, for sure. Yeah. What is interesting is that the work that was done by Jenna, who now has her own lab, but we're collaborating with her through Patrick Pearson lab, what she observed is that she was trying to revert the affinity matured, broadly neutralizing and polyreactive at the same time antibodies back to the germline, so like make them more naive. And in most of the cases, she would lose, of course, the high affinity for the flu targets, but at the same time, she would also lose the polyreactivity. So I think that, there's been hypothesis that there might be like certain, polyreactive germline antibodies that sort of evolved to be more polyreactive. But I think the second case is more likely that this polyreactivity is evolved during the maturation as a way of accommodating the environment rather than being sort of already born with these properties. So I think that the increase from like naive to memory and effector B cells, I think that the increase in the percent of polyreactivity makes sense. It's an evolved feature of the immune repertoire.
And the feature would then be that as your affinity mature, you also target more virus variants or something like that because you're more polyreactive, or yeah.
Yeah. I mean, you can imagine that, antibodies are exposed to many diverse antigens throughout like their maturation process, right? So I'm sure that has an effect on and there is the whole selection that discards the most, with the antibodies with the highest affinity being too dangerous for the system. So there's probably a balance of finding enough support from polyreactive antigens versus the high affinity antigens.
There's a question in the chat. In your heat map for the 338E6 mouse antibody, it looked like a few mutations at the start of the HCDR1 or HCDR2 could abrogate polyreactivity. What do you make of that? Are they somehow disrupting the overall fold of the antibody?
Is this the right slide? Sorry, I cannot read
I,
the question.
I think so. I think so. Yeah.
Yeah, that's the right slide. I think I actually missed what was on the bottom. So you were comparing it to the ones on the bottom, I guess, and showing that it's different for each heat map. Is that the conclusion?
So I'm just showing 3 examples of these single alanine mutations for different clones that come from the same species in this case. And my point here was that I am not seeing any specific patterns in terms of just the heavy chain being involved, which is things that people proposed in the past, that only the heavy chain is involved in polyreactivity, or only specific residues, or only specific positions, or only specific loops are involved in polyreactivity. So the main point here is that, for this clone at the top, the 33.8 E6, it looks like the heavy chain, the light chain is taking the biggest hit in terms of losing the binding for polyreactive antibodies versus the on the bottom. This is an extreme case. The on the right bottom, 41C10 looks like the heavy chain seems to be more involved in the binding with the polyreactive ligands. Is that more or less
Yeah, that answers my question. Thanks.
Okay.
Thank you.
Okay, last question from Anastas, and then we need to close because we're at time.
Thank you, great talk. I've been interested in this problem for a long time. And of course, these conclusions kind of turn some of the understanding, the hypotheses before that on their head. That's not unexpected. What I would expect is that actually you have pretty great heterogeneity in the mechanisms, and we can have both ways. Probably all that is true. Many probably are polyreactive because they are flexible, but some gain polyreactivity by being rigid. So with a couple of examples, you can land in any area of all this heterogeneity. Yeah, and I was thinking in this context, could it be that certain compartments of the repertoire have different rules? So you have studied IgAs here. Then the other, at the end, were they IgA or they were IgGs?
IgGs. Well, actually,
Yes.
the structures, I think I have 2 or 3 IgAs and then 2 IgGs, but then I also included analysis on structures of more IgGs because those are
Yeah, so especially with respect to the selection for polyreactivity in memory antibodies, which could be IgG, that could be one process, and some other would be selected probably in the natural antibodies and IgMs, which are produced by marginal zone B cells, and there maybe we have different mechanisms. That's what I wanted to. So you have never thought of using IgMs, for instance, for this?
No, I do not have access to IgMs. That would be also
that's
because the valency of IgGs, right? They're monovalent versus divalent IgA. You know, pentameric IgMs like that definitely will influence the behavior. That would be excellent. The goal for this study was to have access to these different sources, but
exactly.
It's difficult to have.
Yeah, great talk again. And one thing purely terminologically, do you believe that there exists purely monoreactive antibodies? I think this is an abstraction which is not realistic.
Yeah, I mean, to be honest, like I do have sometimes hard time convincing, you know, kind of like people who think in terms of antibodies, like in an antigen high affinity antigen antibody. Surprised they would just call it stickiness. They would call polyreactivity stickiness. I think that there could be purely monoreactive antibodies, but I think we just don't have the right tools, technology right now to really dig into these low affinity, right? Like even with my direct affinity binding, like I am operating in micromolar affinities, and that's like at the border of detectability with these.
Oh yes, but you can produce a series of epitopes for any antibody, and this is par excellence polyreactivity. I prefer the term polyspecificity, as also Professor Georgiev mentioned that you have certain pattern of reactivity with certain antigens. So polyreactivity leads the thought more into the direction of stickiness, which is an old idea, which is not very useful. So I think that monoreactivity is an abstraction. It's just a gap between the highest affinity and the lowest affinities. Anyway, I was really excited to hear your talk.
Totally agree. Yeah, thank you. Thank you.
And I second that last comment. Yeah, really nice talk. And with that, I thank you again, Marta, for this very nice talk. And thanks everybody for coming. And we have a new seminar coming next month, maybe. Yeah, I think.
In September. The next one's not till September.
September. So that was the last one before the summer. Okay. Yeah. Thanks, everyone. Bye-bye. Bye-bye.